Good morning, everyone. I'm Dr. Ranjini, and I'm going to talk on nursery diseases in coffee and their management. So before talking about diseases of nursery in the coffee, I would like to talk a few things about nursery. So what is nursery? So nursery is a small place where the seedlings or the plants at their early stages are taken utmost care and given optimum conditions for the germination process and subsequently for their growth, and they are taken care in the protected area till they are strong enough to send for the field for planting. And what is the requirement of the nursery? We can sow the seeds in the plant itself. So what makes why we want the nursery? Because the nursery for any plants, any plants are propagated through seeds and by the vegetative propagation to some extent, and they will be very young and they will be sensitive to many harsh conditions. So to protect from the harsh environmental conditions like heavy rains and higher temperatures and heavy winds, this protected environment is needed. And also to some extent to protect the seedlings in their early stages from the wild animals and pests and diseases. So we require the nursery. And what are all the advantages of growing the seedlings in the nursery? Because in the nursery, we can observe each and every seedlings, and we can take care of each and every seedlings in the nursery. And since it is a closed and a small area, it is easy to observe each and every seedlings. And in the nursery level, the seedling growth will be uniform, and when these seedlings are planted in the field, we will get the uniform growth. So and these are all the advantages of nursery. And in nursery, the management of pests and disease also will be very easy. So in coffee, we all know that coffee is a perennial crop, and once planted in the field, it may survive in the field for more than 50 years, and for at least 30 years, it will be productive. So we need to plant a very good quality material. So raising a good quality seedlings take the prime importance in the coffee crop. So coming to coffee nursery includes primary beds wherein it is also called as germination bed, where the seeds are sown and provided with the optimal conditions, that is covering the seed beds with the paddy straw. It will provide the optimal conditions for the germination. And after 45 days, the seedlings attain germination. And once the seedlings attain topi stage, it will be transplanted into the polybasket nursery, or it is also called as secondary nursery. And there it will be taken care up to 6 to 8 months till they are taken into the field for planting. During these phases, from the topi stage, from the germination beds till the seedlings are taken into the field, we face many constraints that hinder the growth of the seedlings. One such is diseases of coffee. So we need to know diseases about the diseases of coffee and their management in order to save our seedlings. So in coffee, we have 3 diseases. One is collar rot disease, and next is stem necrosis and leaf spot disease, and brown eye spot disease. And coming to collar rot disease, it is also called as damping off disease, and it is caused by the fungus Rhizoctonia solani. And this fungus is a soil borne in nature, and it survives in the sclerotial form. See here, you can see the lab grown Rhizoctonia solani, And once the food exhausts from the in the growing plate, it will form a the sclerotial form. The sclerotial form will be mustard-like things, and the same things will be formed in the nursery conditions also. So this sclerotia will be in inactive form, and once they get the favorable condition, they will become active and infect the coffee seedlings. And this fungus infects the coffee seeds and the seedlings at topi stage as well as butterfly stage. And how this disease look like, and what are the symptoms? How can we identify this disease? So this disease occurs in two phases in the seed buds. The two phases are pre-emergence damping off and post-emergent damping off. Pre-emergence damping off means, as the name indicates, before the emergence of the seedlings. Before the emergence, the parts of the seed like embryo, endosperm, and radicle will get infected by the pathogen, and it leads to the rotting of the seed. And then the seed fails to germinate. And in post-emergent damping of the topi stage seedlings, once the seeds get germinated, there will be infection here after the post-emergence. Once after the emergence of the seedling, the infection will be seen. The infection will be seen like a brownish discoloration on the stem just above the ground level. The collar portion of the seedling will get infected. See here, you can see the pictures. The collar portion will get infected, and there will be rotting of the tissues at the collar portion, and finally at the later stages the growing apex will be showing wilting, and it will finally it will cause the death of the seedlings. See, in this picture you can see the clear cut depicting symptoms of the collar rot disease. In the topi stage you can see the rotting of the tissue just above the ground level. The collar portion of the seedlings is rotten. So it is named after that as a collar rot disease. And here also in the butterfly stage also the seedlings get rotten. And what favors this pathogen? The pathogen will be present already in the soil, and it will be present in the inactive form. But some of the factors will boost the pathogen to get activated. So what are all those factors? Normally the pathogen prefers warmer, wet climate, and this disease normally breaks in early summer months. And the most favorable factor is excess soil moisture due to the excess watering, or maybe due to the poor drainage, and also frequent summer showers also favor the disease. And when the disease occurs, when there is overcrowding of the seedlings, it also facilitates the spread of the disease. And next disease is stem necrosis and leaf spot disease. Here, the causal organism of this disease is Myrothecium rhoridum. Here also, the pathogen is soil-borne in nature, and it survives in the soil in conidial form. And this pathogen has a wide host range. It infects many weeds and many horticulture crops. And so many wide host range the pathogen is having. And particularly here in nursery, we have to concentrate on the weeds, because weeds serves as a collateral host for the pathogen, and it will be cause for the spread of the disease. And this disease not only occurs in the nursery, but also in the field condition. The elder plants will get infected. The leaf of the elder plants and the berries of the elder plants in the field condition will get infected, causing leaf spot and berry rot disease in the field. So coming to nursery, this Myrothecium rhoridum infects the seedlings, both in primary seed beds as well as secondary seed beds. There in the collar rot disease, only the primary seed bed, in the primary seed beds will see the infection. But here, both the seedlings in both primary nursery beds and secondary nursery beds will get infected, and in both the cases the stem and the leaves are infected by this pathogen. And coming to symptoms, on the stem we'll see the brownish discoloration on the tender stem. And here on the myrothecium, we can see more than one lesion in the single stem. And at the later stage, we can see the cushion-shaped black fruiting bodies surrounded by the white mycelia. So we can see here, we can see in the first picture, it is marked yellow, and we can see the fruiting bodies. We can for the naked eye only, we can see the fruiting bodies. And here, when the fungus infects the stem, the seedlings either the breakage of the stem will occur, or it start wilting and it will die. Here, if the pathogen infects the stem, we can't take any measure. Means we will lose that seedling, that particular seedlings. And here we can see in a clear manner that the difference between the collar rod disease and the stem necrosis disease. As here only the collar portion of the stem is infected, but whereas in the myrothecium roridum, see here two spots are there. Two necrotic lesions are there on the stem. Here in the elder seedlings, the middle portion of the stem is infected, and in the elder seedling, the lower collar portion of the stem is infected. And this is the larger view of the spodocia, or the fruiting bodies of the myrothecium roridum. Here, this is the presence of fruiting bodies is the clear difference between the collar rot and myrothecium roridum, because myrothecium roridum can infect the collar portion also, and some may get confused with the collar rot and the myrothecium roridum. So presence of fruiting bodies will indicate the infection of the myrothecium roridum. And on leaves, here also both in the primary beds and the secondary beds, the infection takes place. And here you can see the tip apex portion is also getting infected. The leaves and the stem both are infected, and on the cotyledonary leaves, the spots can be seen. Here, the spots will be initially water soaked, and they will spread, forming brown color, brown concentric rings, and they will sometimes they will cover the entire leaf surface. And black fruiting bodies are noticed on the corresponding lower surface of the leaf all along the concentric rings. The fruiting bodies will be arranged concentrically when you keenly observe the lower surface of the infected leaves. And in severe conditions, 2 to 3 such spots. Here you can see in the first picture, 2 to 3 such spots will join together. See here it is joining, and they join together and cover the entire lamina of the leaf. And occasionally, when there is too much rain, the center of the lesion cracks and irregularly ruptures. And sometimes the leaves won't fall, but stand and here you can see the whole seedling is getting infected, and it is fully loaded with the fruiting bodies of the pathogen. It is the larger view of the fruiting bodies on the leaves. And what favors this pathogen? Prolonged wetness of the plant parts, particularly when there is continuous and a very heavy rains. When there is continuous wetness of the leaves, the disease will be seen in the severe form, and it will spread rapidly. And also, due to the heavy rains, the low temperature and high relative humidity also favors the pathogen, and excess shade in the nursery, and of course, overcrowding of the seedlings. Once the healthy seedlings will come in contact with the infected ones, they will get the infection, and it will spread rapidly when the conditions are favorable. And coming to the management of the collar rot disease and stem necrosis and leaf spot disease, and in cultural management, we can avoid primary nursery beds, the sowing of the seeds in the same location for 2 to 3 seasons. This is because we can reduce the pathogen load. If the seed beds are placed in a same location, the pathogen load may be more, and they will be present in the inactive form. So it is better to change the primary nursery beds once in 2 to 3 seasons. And always use sun-dried sieved jungle soil compost and sand in the recommended proportion, that is 6:2:1. And also avoid excess watering to the seedlings. As I told, dampness will cause the it is a favorable condition for the pathogens, both the pathogens, the Myrothecium rhodotum and the as well as collar rot pathogen. So avoid excess watering in the nursery, and also maintain good drainage, since the poor drainage may cause dampness in the seed beds. And also cleanliness. Why cleanliness is important here? I told Myrothecium rhodotum infect the weeds. So the weeds may harbor the pathogen and it may spread the disease to the seedlings. So please keep the nursery clean. And in chemical management, always use fungicide treated seeds. If the seeds are procured from our institute, from CCRI, it will be fungicide treated. If you are taking the seeds from other source, please ensure it is treated with the carbendazim 50 WP at 1 kg per 1 gram per kg of the seed. So, and before imposing any fungicide to the seed beds or the secondary beds, please remove the affected seedlings or the affected parts and destroy them. If you don't destroy it, if you throw them nearby place in the nursery, the winds and the rain may carry the spores of the fungus and it may spread to the seedlings. So please destroy the affected parts of the affected seedlings. And then drenching of the fungicides in the seed beds, like carbendazim 50 WP at 1 gram per liter and mancozeb at 2 gram per liter alternatively to the seed bed once in 15 days will manage both collar rot as well as Myrothecium rot in the seed beds. So this drenching must be done using the rose cans. And in the secondary beds, for the management of stem necrosis and leaf spot disease in the secondary nursery, foliar spray of fungicide propiconazole, or it is called as tilt, at 1 ml per liter, and mancozeb 75 WP at 2 gram per liter may be sprayed alternatively once in 20 days. Whereas here you should take care that the fungicide is covered both on the upper surface and the lower surface of the leaves. And you should also take care, continuous use of the propiconazole must be avoided, because continuous use of propiconazole may cause crinkling and cupping of the leaves in the nursery. So we recommend the alternative spray of propiconazole. It is a systemic fungicide, and mancozeb, this is the contact fungicide. So we recommend alternative spraying of the systemic as well as contact fungicide once in 20 days. And not only the propiconazole, but our recent studies, in our recent studies, we have found out that the fungicides Prioxur, Opera, and Nativo can be used in the lower concentration, that is 0.5 ml per liter, and in case of Nativo, 0.5 gram per liter can be used for the effective management of the stem necrosis and leaf spot pathogen. But here also one should take care that the concentration must be in the lower doses. It must not go higher than 0.5 ml because it may cause toxicity to the seedlings. So always be better to use a systemic fungicide alternative with the contact fungicide, that is mancozeb 2 gram per liter, for the management of stem necrosis and leaf spot disease. And next is the brown eye spot disease, and the last disease. It is caused by the fungus Cercospora coffeecola, and here also the fungus survives in the plant debris in soil, and this pathogen also this pathogen also infects many hosts in the nursery level. So many of the weeds which are grown in and around the nursery are infected by this pathogen. And infected seedlings show weak and stunted growth. Here the seedling won't die, but it shows weak and stunted growth and become unfit for the planting in the field condition. And when coming to symptoms, here there will be the infection starts as necrotic spots which are brown in color and surrounded by a yellow halo. There may be multiple spots in each leaf, and sometimes there may be a single spot, and this yellow halo, the presence of yellow halo is due to the production of a toxin called cercosporin by the pathogen. On severity, here you can see in the third picture, the leaf turns yellow and falls prematurely. And due to the reduction in the photosynthetic area, the seedlings cannot produce food, and they may not attain desired height to plant it in the field conditions. So this disease not only occur in the seedling stage, but also once the seedlings, when they are planted in the field conditions, during the first year of the planting, that particularly the seedling will be infected by this pathogen. And the favorable factors, as I told, direct exposure of the seedlings to the sunlight, particularly in the months of April to June and September to November, when there is direct exposure of the seedlings to sunlight and insufficient overhead shade in the field conditions. If the top canopy is not sufficient, it may be favors the disease. And also some reports say the nitrogen deficiency also favor the disease development. And here this slide shows the difference between the leaf spot of myrothecium and leaf spot of Cercospora coffeecola. Here in brown eye spot disease, you can see clear cut yellow halo around the spot, and here there will be no concentric rings. Whereas in myrothecium leaf spot, it will be little darker, and it will be in the concentric form. And coming to management, here as I told, maintenance of required filtered shade is required it is provided by providing the 50% poly shade net, and in the field condition protect the seedlings by providing hutting, and also by providing the good overhead shade. And if this disease is noticed in the nursery level as well as field level, the fungicide tubiconazole 25 EC at 1 ml per liter can be sprayed once in 20 days. And in the field condition, you can also use Bordeaux mixture at 0.5% for the management of the disease. And I would like to discuss some health tips for maintaining a good nursery. So providing good drainage to facilitate the easy flow of water, as the dampness will provide the favorable conditions for the multiplication of pathogens. And regular tracing is required, because if disease is not noticed for few days, it may and if the condition of condition are favorable for the pathogen, it may destroy many of the seedlings in the nursery. And in seed beds, the collar rot pathogen may destroy the whole bed if the regular tracing is not there. And regular spray schedule has to be followed. If the disease is there or not, regular spray schedule has to be followed to maintain the health of the seedlings. And maintenance of sanitation. So weeds are the collateral hosts for the pathogens in the nursery. So maintenance of sanitation in and around the nursery is a must. And these weeds compete for the nutrients with the coffee seedlings. So sanitation is important in the nursery. And cover the seedlings by rainout shelters during continuous heavy rains. As I told, the main favorable factor for Myrothecium roridum is continuous wetness of the leaves. So by covering the seedlings by rainout shelters during continuous heavy rains, we can avoid this disease. And exposure of nursery site and the components of nursery mixture to the sunlight helps to reduce the inoculum of the pathogen. And This is the picture of a rainout shelter. One should take care that the seedlings must not be fully covered. It must be half covered, and good aeration must be there. And this rainout shelter will be helpful um, from the continuous wetness of the seedlings, and also protect the seedlings from rain splashes, because rain splashes sometimes carry the pathogen from the soil to the seedlings. So if you install the rainout shelters during the continuous rains, 50% of the stem necrosis and leaf spot disease we can avoid in the nursery. So thank you.